Good morning, class. I'm sorry I'm not here today. My little boy is running a fever of about 103, so I'm taking him to the doctor today. However, we are going to have our test on Monday, and today I'm going to use this nifty software to go through your lesson for the day. So, please pause the video and get out your Dihybrid Cross worksheets that I gave you yesterday. They look like this. Practice problems for dihybrid crosses. Okay, now that everyone has those out, I'm going to use the inner right space here to go over the answers. So let's get started. Number 1, a pea plant is homozygous for the genes purple flowers and round seeds. Using the letters P and R, write a genotype for this plant. Now, in dihybrid crosses, remember that there are going to be 4 letters. We're going to have genes for the purple color, and we're going to have genes for the round seeds. It's homozygous for purple, and up here it says, for the following problems, assume that purple is dominant over white and round is dominant over wrinkled. So, for our dominant purple flowers, we're going to put a capital P and a capital P because it says it's homozygous for the genes purple flowers. It also says it's homozygous for the genes round seeds. And round is dominant over wrinkled, so we're going to have 2 capital R's. There we go. We don't need another P. So 2 capital R's. Number 2, what kind of gametes can this pea plant produce? This question is asking you to do the FOIL method that we talked about yesterday. So, for the FOIL method, we're going to use the first letters, so P and R. Those are the first of the 2 different kinds of letters. So one gamete can be PR. We're going to use the outer letters, 1, 2 here, big P, big R. That's the second gamete. Inner, the capital P and capital R here, and the inside, capital P, capital R, and then the last letters, so the last P and the last R. P and R. That one was pretty simple. I hope you see the pattern that since all of the letters were capital, in our gametes we'll have all capital letters as well. So let's go down to number 3. Write the genotype for the following plants. A plant that is heterozygous for the color gene and has wrinkled seeds. So let's do the color gene, our P's, and it's heterozygous, which means it's going to have a capital P and a lowercase p. It also has wrinkled seeds, so we're going to do 2 little r's, because wrinkled is recessive. B, a plant that has white flowers and wrinkled seeds. White flowers are recessive, so we're going to have 2 little p's and 2 little r's. And a plant that is heterozygous for both genes. So we'll need a capital P and a lowercase p to be heterozygous for color, and then a capital R and a lowercase r to be heterozygous for the shape of the seeds. Okay, let's go down and actually do our Punnett square. Let's see, I'm going to insert a table. This will be easier. We need okay. Wow, they don't need to be numbered, do they? Actually, this was let's make this look more like a Punnett square. Here we go. I'm going to put the parents in these top boxes just to make it easier, but let's figure out what the parents are for this cross. We're going to do the FOIL method again up here. So we have to do two separate parents. Let's start with this one. Do the first letters, capital P, capital R, outer, capital P, lowercase r, inner, a little p and a big R, and last, a little p and a little r. Alright, so that's our first parent. We're going to put those down here. P, R, oops, P, R, little p. I'm going to put the tail on my little p there. Big R, little p, little r. Alright, now let's do the second parent up here. And they're all little letters, so that helps. Let's go through the FOIL method anyway. First P, first R, P, R. Outer P and outer R, P, R. Let's put the tails on my little p's so I know the difference. Inner, little p, little r. And last, the last p and the last r. We will put those over here as well. Little p, little r, little p, little r, little p, little r, little p, little r. Okay, I'm going to switch back to using the keyboard, and we're going to fill this Punnett square in. Actually, yeah. So, we had big P, big R, big P, little r, little p, big R, and little p, little r. And then all of these, Okay, and as we fill these in, we're going to bring these capital P's down. We're going to bring these capital P's down. Now we're going to bring these small p's down. Have to make sure they stay small p's. And we're going to bring these small p's down. Okay, so we've brought these p's down. Let's bring these over. We have a little one, little, little. In fact, they're all going to be little, so we're going to go ahead and fill those in. Alright, now we need to fill in our R's. We're going to bring this big R down. And now we're going to bring this little r down. Another big R. And a little r. And all of these are small, so we're going to bring those over. And we will have filled in our Punnett square. And that's all that number 4 asks you to do. So there's our completed Punnett square. Number 5 was a bit confusing, yes? Hopefully I talked to most of you in class about this one. We have a fruit fly with normal wings, and this is considered the wild type, meaning this is what you're going to find in the wild most often. Whenever you hear wild type, that just means that it's showing the dominant traits, because that's what you would find in the wild. So normal wings are dominant over vestigial wings, and brown bodies are dominant over ebony bodies. A wild type fly, and then let's separate this out here, whose mother was ebony and vestigial. So we're talking in letter A, we're talking about this wild type fly. Because he's wild type, we know he's showing the dominant N for normal wings and big B for brown body. Okay? But it tells us his mother was ebony and vestigial. So if the mother had vestigial wings, she was homozygous recessive, which means she had to pass on the recessive trait to this wild type fly. So here is our wild type fly. That's the answer to A. So what would the genotype of an ebony vestigial fly be? Ebony is recessive to brown, so we know that they're going to have 2 little b's. Vestigial is recessive to normal, so we're going to have 2 little n's. It says list the phenotypes and the genotypes of the cross in the space below. Some of you picked up on this, so what we're actually doing is we're crossing these 2 together. And some of you picked up on the fact that these were the same as these, just with different letters. So we have, if we do the FOIL method with this, the offspring, the gametes, can be only little b, little n, little b, little n. Okay? And the parents up here, if we do the FOIL method again, we're going to have big N, big B for the first ones. Outer ones, big N, little b. Inner, little n, big B. And last, little n, little b. So there's the makings of our Punnett square. It's just the same as this. So you could actually, if you're willing to substitute letters, this actually shows the same information as this. So once you fill it all in, it says list the genotypes and the phenotypes of the cross. So let's go ahead and fill this in. I think this might be faster than the other way. Now, we know that all of these are the same, right? So really, all we need to do is cross the one at the top. This is much like the example in our notes from yesterday, which are online. You can look them up and listen to them again. So here we go. There's our homozygous recessive. So genotypes, we've got N. So there's one. This is this column. And what is the phenotype of this one? Well, it's wild type. So this one is, sorry about my writing, normal and brown, right? So that's one phenotype and one genotype. Then we have normal and ebony. So there's that one, and it will cover those. This one is going to be vestigial and brown. And the very last one is recessive for both traits. So it will be vestigial and ebony. Remember, these are the phenotypes. We have 4 different looking flies. These are the genotypes. Okay? Alright, if you have any questions, please write them down on your paper and email me this weekend. Remember, our test is still on Monday. You will not have to draw one of these on the test. You will be given something like this with all of the offspring blank, and you need to know how to pull the letters down and across. But you won't be required to do the FOIL method on the test.